Hi everyone. So today I'm going to be talking about Autophagosome ATG5 and how it's related to the KRAS oncogene. Now, autophagy is a process that we talked a little bit about during class. It's the catabolic process through which the cell will essentially degrade its own organelles. It will do this by forming this membrane around the organelles and whatever the cell is trying to degrade. And this membrane will then form a vesicle called an autophagosome, which will then fuse with a lysosome, and the lysosome will degrade all the cellular components inside of the autophagosome for its nutrients or whatever. This is a normal cell process. The cell will do this usually in situations with a stressful environment, such as hypoxia, starvation, something like that. Even though this is a normal cellular process, it does have certain adverse effects that we see on cells in the body. We see these effects, especially in aging and neurodegenerative diseases. It also has effects on several different types of cancer, such as colon or pancreatic cancer. Now, going a little bit more in depth into this process, we have these, we have 20 or 20 plus core proteins that are responsible for the formation of these autophagosomes. These are called ATG proteins. As you can see here down at the bottom of the screen, we have this cascade of different proteins. We see ATG10, ATG7, ATG5 specifically, and all these are important for the formation of these autophagosome vesicles, which encapsulate the organelles, which will then be degraded by the lysosomes. Now, specifically, for our purposes, autophagy is regulated by many things, but the RAS pathway is one of the most prevalent regulators of this. We talked about the RAS pathway quite extensively during class. It's a signaling pathway that is not just used for regulating autophagy, it's used for regulating many different pathways throughout the cell. It is a receptor tyrosine kinase that whenever a growth factor or anything like that binds to these receptors, it'll phosphorylate GRAB2, which will then activate SOS, then the RAS protein. And then after that, it'll activate a whole cascade of different kinases, such as the RAF kinase, MET kinase, ERK kinase. The ERK kinase specifically has a lot of branching things off of it. So the ERK pathway specifically is important for regulating autophagy. Different things will regulate autophagy, but the ERK pathway is one of the most prevalent ways of regulating this. And this pathway is found in pretty much every cell throughout the body. Different cells will have different effects to this pathway being activated. Kind of just depends on the cell. But the RAS pathway is a good pathway to study for us because it's prevalent in pretty much every cell. Now, there can be aberrant effects to having this pathway mutated or changed, and one of these is the KRAS oncogene. We talked about this a little bit during class as well, but the KRAS oncogene is essentially when the RAS protein I talked about earlier is a little bit overactivated. So it's turned on a little bit more than it should, so it's activating things in the pathway that it shouldn't, and this could essentially have effects on many different things, but it will also affect autophagy, which is what we're looking at. KRAS stands for Christian rat sarcoma viral oncogene homolog. Because this pathway is present in many different cells, we would expect to see that this oncogene would also be present in many different types of tumors, which is the case. It's present in 25% of all human tumors, but especially we see this oncogene present in colon, pancreatic, and lung carcinomas. The gene for this oncogene is found on chromosome 12. Now, why did we choose this research? So whenever we were researching this and the effects that the KRAS oncogene may have on different things, for autophagy, just looking at, we assumed that autophagy would have a negative impact on tumor growth because autophagy is just a cellular process where it degrades things, it degrades your organelles and different things within the cell, we would expect that this cell could use autophagy as a defense mechanism against tumor formation and tumor growth. And we did see this in the studies that we saw in early stages of tumor growth. We saw that autophagy was used as a tumor suppressor. It prevented the growth of these tumors while they were still in their early stages. However, as we continued looking into this, we saw that in advanced stages of tumor growth, when the tumor was able to surpass all of the cell's mechanisms, preventing its growth and such like that, the tumors would use autophagy as a way to increase its growth. It used it, the autophagy wasn't used to suppress it. In fact, it was used to enhance its growth and push it forward. And we were really curious, you know, like how does the tumor cell do that? You'd think that using a way that the cell would use to destroy the tumor, why would the tumor use that in order to enhance its growth, you know? So specifically, we were looking at with autophagy, what specific part of autophagy were these tumors using? So we looked through and we determined that ATG5 is a very important core protein for autophagy. So we wanted to see the effects that ATG5 specifically has on this KRAS oncogene, on this tumor growth, on essentially if ATG5 has any specific impact on tumor growth.
Okay, so now that we have a better understanding of some of the background information, we're going to be moving on to looking at the two different aims of our study. The first goal of this study is to look at the relationship between ATG5 and the KRAS oncogene. By examining this relationship, we'll be able to identify if this oncogene has any adverse effects in cancers that use the ATG5 to upregulate autophagy. As Joe said earlier, autophagy can be used to enhance tumor growth, and having better understanding of this type of relationship can lead to understanding these types of pathways better. Also, there has not been as much research done specifically with the KRAS oncogene in relationship to the ATG5, so our study is trying to take a closer look into that. The second goal of our study is to see if there's a correlation between the upregulation of ATG5 and cell death. So, if more of the KRAS oncogene is being expressed, this could lead to an increase in the production of ATG5 protein, which could then lead to the upregulation of autophagy. With this upregulation of ATG5, we expect to see an increase in autophagy. The goals of this experiment is basically to help advance this area study, and this could lead to an overall better understanding of the KRAS oncogene and its role in autophagy and cancer tumors. So now that we understand where we're going with this study, with the aims, we're going to look into our hypothesis. So we're interested here in the addition of the KRAS oncogene to a fibroblast cell line, which will change the levels of ATG5 in the cell. So we expect with the addition of the KRAS oncogene to increase levels of ATG5 in the cell. And we believe this because the cell will attempt to use autophagy as a mechanism to inhibit the growth of the cancer tumor. We'll be using various lab techniques that we've talked about in lecture and in lab throughout the year, and we'll be using those to test our hypothesis, which leads us to basically our next slide, which will give an overview of our study. So this slide is basically showing that our study is broken down to 5 basic pieces. The first part is to obtain a fibroblast cell line from mice, and after we have those cells, we will infect the experimental group with the KRAS oncogene by a retrovirus and our control group will not have the oncogene inserted into them. And we'll be looking at those two cell lines. Next, we'll be using quantitative PCR, basically to ensure that the oncogene is present in the cells that we want it to, and we can begin to study those cells. Following this, we will be using flow cytometry, and this is used in our study to quantify cell death and to see the differences in the experimental and control groups. Finally, we'll be using a Western blot to compare the levels of ATG5 that is found in the two groups. So this is just a quick overview of what our study will be looking like, but now Nolan will be going into more of the specific details regarding the certain tests that we'll be running in our experiment, as well as the results that we are expecting to see.
Okay, so hi guys. As Madison said, we are going to test the fibroblast cell line, and we're going to give those fibroblasts KRAS by retrovirus. The cells given KRAS by retrovirus is going to be experimental. Our control is just going to be our regular fibroblast cell lines. And so to make sure that the retrovirus was successful in inserting the genes into the fibroblast cell lines, we are going to use quantitative PCR to do this. We're very familiar with quantitative PCR, and this is just done to double check. As you know, a lot of papers will double check their findings just to make sure. And we are injecting these fibroblast cells with KRAS, so we're going to expect that some of these cells are going to die. And to quantify this cell death, we could use propydium iodide. As we know, as the cell degrades, the cell membrane is going to degrade as well. As the cell dies, the cell membrane is going to degrade. And this will allow propydium iodide to enter into the cell, and we are able to sort out the live cells from the ones that have propydium iodide inside the cell. And so we're going to look at these live cells and see the expression of ATG5 in them. And by doing that, we are going to use Western blot. We're very familiar with how Western blot works. We're going to separate it with the gel and transfer it over, use antibodies for it. And we're going to expect to see a higher expression of ATG5 in the cells given KRAS, and very minimal or none in the control. We could use ERK just to make sure, as a control, to make sure that the experiment worked correctly, because as we know, ERK is everywhere in the cell. It's very commonly used. And so we're going to expect a high expression of KRAS in these, of ATG5 in our experimental cells. And so that's our experiment. Now we're just going to go on to some critiques. So if the experiment didn't work out due to all of the fibroblasts dying, we're going to take a known KRAS cancer and test for the same exact thing. So look for ATG5 in the cells. And the mechanism for why autophagy promotes tumor growth in the late stages of tumor progression is not very well known. So we can look into that. If there's no correlation between ATG5 and KRAS, then we could look at the other proteins known to be involved with RAS oncogenes. And if the ATG5 levels do correlate, further studies would need to be performed in order to confirm ATG5 is the protein responsible for that correlation. So in conclusion, if the hypothesis is proven, this could help with the development of drugs to target ATG5 in the KRAS cancer, in cancers known to have KRAS. So if ATG5 is an autophagosome with critical importance to the KRAS oncogene, one of the mitigating effects of the KRAS oncogene would be downregulating ATG5 levels in the cell. So this research could open up new avenues of tumor treatment and recognition in the cell by finding correlations between different tumors and the proteins upregulated by them.
Thank you so much for listening, guys. Here's just a list of our sources that we used. Thank you.